Welcome everybody to this week's mini seminars in the working group on the multi-scale modeling and viral pandemics. Today we've got two mini seminars scheduled and possibly a little bit of discussion time afterwards on a few items of business. I remind you that the meeting is being live streamed on YouTube and that recordings will be made publicly available. So please remember that. We have, as usual, myself, Reinhard Lavenbacher, the co-lead on the working group, web administration by Jim Sluka, who's on the call, and Bruce Shapiro, whom all of you know well from your communications. As usual, I'll remind you we have a Slack channel. You're invited to use it. We do need people to bootstrap it if it's going to be helpful. If you have any issues getting onto it, contact us and we'll do our best to help you out. There's also the IMAG MSM Wiki page. We'd love to have you post your material, update it, and help people make it an interesting website. I was going to change the order of the slides a little bit today, just to remind you for our upcoming meetings and mini seminars on the 28th. We have two teams from France and Luxembourg talking about cooperative modeling related to COVID-19 infection. They've done some very interesting crowdsourcing and collaborative modeling development. And then on February 4th, we have Morgan Craig talking about modeling of immunological responses, and Greg Forrest talking about viral transport. As usual, please suggest potential speakers that you'd be interested in. Please volunteer if you'd like to speak and help us keep this lively and interesting set of seminars. With that, I don't want to take any more of our speakers' time. We're going to begin with typo on your name. I'm sorry. No, typo not. You're okay. Seminar by Kevin James, Department of Biomedical Engineering, University of Virginia, complete kinetic modeling of an RNA pathogen. And I'll turn it back to you for the screen share.
Can I get a confirmation that everything looks okay? Okay, I see you nodding. All right, thanks very much for the introduction and the opportunity to speak in front of this working group. I only have 15 minutes, and so we'll get right down to it. Overall, my laboratory is interested in gaining a systems level understanding for how mammalian cells respond to complex perturbations. And the reason why is that we think it's fertile ground for iterating between experimental manipulations and measurements, computational models, and analytical data mining. A lot of the work that my lab does relates to cancer, where we are interested in gaining a sense of how cells adapt to that first handful of mutations or chromosomal changes during premalignancy. However, over the past 5 or 10 years, we've become equally enthusiastic about adopting a similar perspective for host cell responses during viral infection. And if we're going to fully embrace such an approach, that iteration between measurements, mining, modeling, manipulation, I'd like to make the case for enteroviruses as a general testbed and for the enterovirus Coxsackievirus B3 as a leading test case. I think it was Einstein that said everything should be made as simple as possible, but no simpler. And in the viral world, enteroviruses are about as simple as they come. They encode a single-stranded RNA genome that yields one translated polypeptide. And what that means is each translation event will yield exactly one copy of each of the mature proteins shown here in blue, green, and red. And that's an important stoichiometric constraint for modeling. Moreover, we have quite a good understanding of what all these mature proteins do for the viral life cycle, meaning that we can organize and abstract them in an authentic way. Third advantage is that enteroviruses have decades of quantitative enzymology, genetics, and biophysics available. That information is scattered all over the literature, but it's there to mine. I highlight this remarkable paper that titled "The Absolute Number of Infectious Enteroviruses Released from Single Cells," so a single cell assay nearly 60 years old. And last, enteroviruses cause human disease. And while some enteroviruses, such as poliovirus, has been called a research field with a rich past but no future because it's nearly eradicated from the globe, these other strains listed are still active and infect people across the globe. The biggest void we saw in enterovirus research was the scarcity of cell scale models. There were a few instances of models for evolutionary dynamics, but nothing related to the viral life cycle itself. And because the genetic architecture of enteroviruses is so simple, because there are 70+ years of knowledge to build upon, and because these viruses act quickly, we sought to attempt something audacious, and that was to build a kinetic model that was complete from two standpoints. First, in covering the temporal arc of infection from very start to the end. And then two, complete from the standpoint of the stoichiometry of components needed for the infection to take place. And that relates to that single-stranded genome and the single translated polypeptide. So what it's showing schematically here is the modular architecture of the model that we built. And I want to walk you through the design and the life cycle of enteroviruses as it's currently understood. All right, so step one here is that the virus docks on membrane receptors, is internalized, and then this positive strand RNA virus, RNA genome is delivered into the cytoplasm. The delivered positive strand RNA is used as a template for translation using ribosomes from the host cell. And this single polyprotein is autocatalytically cleaved into mature protein subunits with direct roles in the life cycle. Down here, we have an RNA-dependent RNA polymerase that synthesizes negative strand complementary to the positive strand, and then uses this negative strand as template to make many copies of the positive strand for more translation and eventual virion formation. The enteroviral proteinases 2A and 3C are required for polyprotein cleavage and maturation. And they also help steal more ribosomes from the cell by cleaving host cell targets. And then the structural capsid proteins instantaneously get together. They form 1/5 of a self-assembled pentamer, and 12 of those join around a viral RNA to form the mature encapsidated virion. At the center of it all are a set of host cell feedbacks that give rise to an interesting systems level problem. Viral replication requires double-strand intermediates during this replication step here that are sensed and give rise to the induction of hundreds of so-called interferon-stimulated genes, which shut down the life cycle at the points indicated, here, here, and here. The pathway for these ISGs is itself damaged by viral proteinases, creating a kinetic competition between the microbe and its host. Overall, the design of this model had an architecture that we designed to be modular, recognizing that while delivery up here is very specific to each enterovirus, the intracellular core, all the downstream processes, is shared by all. And what we hope this would do is open up opportunities to repurpose this Coxsackievirus B3 model for other enteroviruses down the road. I want to give you some highlights of the model refinement and then transition to the central message of this work. The first thing that we learned in trying to construct this model was the importance of bookkeeping. One complication of trying to build a model, quantitative virologic model, is that not every virion released by a cell is infectious as defined by the ability of that virion to kill a field of susceptible host cells, so-called plaque-forming unit. And the ratio of viral particles to infectious PFUs is large, and more bedevilingly for enterovirus is highly variable, even within the same strain of virus. It's representative particle to PFU ratios on the right. Specifically, poliovirus is as close as can be to CVB3, but from a genetic standpoint, but with such an enormous range of ratios here, it was virtually impossible to put the experiments that we were doing and trying to reconcile with the model and put them on the same scale as the computational simulations. But what we did during some intense mining of our own experimental data in the thick of COVID quarantine, we got a ratio estimate for our preparations of Coxsackievirus B3. And we were then able to reconcile our quantitative data of the dynamics of positive strand RNA genomes, negative strand RNA genomes, and those structural capsid proteins with the model simulation. To orient you for the rest of the talk, what I'm going to show are model results here in blue, or in maybe other colors in the slides, is the median plus or minus a 90% interval defined by 100 different simulations using parameters randomly sampled about a coefficient of variation, 5% of all the parameters, that best estimate that we drew from either the literature or from the biology. And then overall, this model that we constructed is about 55 ordinary differential equations, roughly the same number of parameters. And of those parameters, about 90+% are constrained, not fitted, fixed, either from the literature values or from our own experiments that we collected in the lab. One of the things that we needed to come to terms with, though, with this model is that even though it was very detailed in many respects, there were some parameters that were and forever would be unknowable. And those mostly revolved around these interferon stimulated genes and their connectivity in the middle here. So let's pull the veil off of this part of the cartoon. As I said, this model is built from differential equations, and the way that we define this edge here is a maximum conceivable rate of interferon stimulated gene synthesis that we then elaborate with two terms. The first abstracts a 9-step biochemical mechanism into a sigmoidal input-output equation with two free parameters. What I'll note here is that this double-strand RNA concentration is something we take directly from the model stimulations. And then we have a second term here that encodes the negative feedback, that antagonism by the viral proteinases. We also take the concentration of those proteinases directly from the model, but the potency of those proteinases and the lumping of their substrates is something that we will never fully know in vivo. And all of that is really embedded in this EC50 effective concentration for how much negative feedback you get as a result of how much proteinases are in cells at the time. But even with this uncertainty, we did have some important qualitative constraints based upon experiments. And so there are a couple of scenarios that we needed these lumped parameters to conform to align with the biology. First was that if there was no double-strand RNA sensing in the model, should allow the infection to take off and proceed unabated. Here what I'm showing model outputs now of the virions, released virions, plus a rough estimate of the number of PFUs, plaque-forming units, according to that ratio that I described from before. If we put in double-strand RNA sensing alone, the endogenously triggered interferon-stimulated response is sufficient to stop the infection. But because that pathway is ordinarily damaged by those viral proteinases, the infection proceeds with kinetics that are only slightly delayed when from when antagonism is in place. However, we can also run the model to receive interferons exogenously. This would be akin to paracrine stimulation from infected neighbors or immune cells. And we took this in the model as going straight to maximum synthesis in the model. And if that's done right at the time, artificially right at the time when the virion docks on the plasma membrane, infection is completely averted. However, if we wait too long to add that interferon stimulus, the warning comes too late, the negative feedback is already engaged, and the infection proceeds as if they had never received that interferon stimulus. So together, these qualitative constraints turned out to be strict criteria for the range of acceptable values for model parameters, these lumped parameters that were phenomenological. Now, on this last slide and this one here, I discussed how the model was, if you will, wrong in some respects, but I didn't talk about whether it was useful or not, which was in the title. And so let's apply the model and see what we can find. And the way that we did this was by sweeping through all of those phenomenological parameters, really tackling them head on, iterating through hundreds of different parameter combinations, about 4,000 or 5,000 different model simulations, with or without neighboring interferon stimulation added to the simulations at different time points post-infection. And in doing so, we found something interesting and non-obvious when we changed the susceptibility of the double-strand RNA pathway to damage by viral proteinases. And to remind, that was that EC50 value in the negative feedback that I described one slide ago. If we just altered the model to render the cells mildly more resistant, we saw this anticipated mild decrease in virions release, turning down that negative feedback. However, when we ran the same simulations with interferons signaling from neighbors, the behavior changed, depending upon the time at which the interferon was added. At early times, there was a dampening of both responses, shown here in 4 hours, and a convergence, if you will, of the difference between the standing model and this resistant model. But as the interferon was added in a more time-delayed manner, we saw a more sustained responsiveness to the paracrine interferon only in the resistant case, whereas the base model had returned close to the situation in which they had never added interferon before. And so this was on the border of something that we could test experimentally, but to be able to do that, we needed to dive into this 9-member pathway and identify precisely where the viral proteinases might be targeting and how. And so long story short, using clues from the literature, we converged upon the mitochondrial antiviral signaling protein MAVS, which is reported to be targeted by the 3C proteinase and enteroviral proteinase. And we found that MAVS was indeed cleaved as a 35 kilodalton product here in our host cells when they were infected with CVB3. But we needed to do better because the only way to make the pathway resistant was to know the exact site on MAVS that was cleaved. And here we turn to mining through bioinformatics. MAVS is a very interesting protein. It is, Harmit Malik's group showed that the gene is rapidly evolving and in primates, and its sequence may reflect past exposure to ancient viruses. And if one looks at the MAVS sequence in primates globally, there's a splice site variant that adds 6 amino acids to the protein sequence in hominoids and old world monkeys here on the top. It's absent from new world monkeys on the bottom. And in that sequence is a strong match for the peptide motif that enteroviral 3C proteinases like to cut. And this was reconstructed by my lab manager during the quarantine. Importantly, chimpanzees, us, and even African green monkeys here in green show evidence of Coxsackieviral infections in the literature. But New World monkeys, such as the marmoset, are confirmed negative even when those marmosets are maintained in captivity. And so from here, the experimental path to testing this model prediction was pretty clear. We wanted to target this candidate cleavage site and then evaluate its effect on infectivity. And that's what we did. We engineered cells to contain a doxycycline-inducible flag-tagged MAVS with or without that position 271, that splice site addition, mutated from a glutamine, which is absolutely required for the proteinase activity, to an alanine, which would destroy the site for 3C proteinase. We made the inducible expression pretty strong to dilute out the impact of endogenous MAVS. We confirmed that CVB3-induced cleavage is significantly reduced in the alanine mutant compared to the glutamine, although the overall effect is incomplete because of the residual cleavage of endogenous MAVS. And we showed that the difference in cleavage coincided with a difference in viral plaques and infectious virions released after infection with CVB3. Now for the test. We infected these cells with CVB3, stimulated them with recombinant interferon added into the dish at several times post-infection. And what we found was that at early times, in that 4 to 5 hour window, like I showed in the simulations before here, both lines showed reduced infectivity. But at later times, the effective interferon was lost in the wild type case, whereas the resistant line remained less infections. And so collectively, we think that the so-called wrong parts of the model were useful in uncovering this nonlinearity, this interaction between the timing of paracrine interferon and the resistance of host cells to antiviral antagonism. These findings got us more interested in MAVS regulation and the history of the various synonyms that it goes by. This figure on the left is structurally accurate, but it's somewhat complicated. So I'm just going to break down the acronyms into sensors, transducers, and effectors. What occurs here is that you have double-strand RNA forming filaments on the mitochondrial surface. They go on and polymerize MAVS, which is normally mitochondrial localized here through these domains that bind to the double-strand RNA. And it's that oligomerization of MAVS that gives rise to signaling downstream in the induction of interferon stimulated genes. There are many different groups that have cloned MAVS, and there are some discrepancies about where those MAVS versions that people use came from. And the most popular one, which was the one that we started with, is not the reference protein sequence for MAVS. This was discovered by my research scientists in the lab. And the difference here, to be specific, relates to a glutamine in position 93, it's highlighted here, whereas the reference human proteome has a glutamine. And so what I want to orient you here is that this is the flag tag construct that we used for the experiments before. This is the self-assembly domain that eventually links to double-strand RNA. This is the mitochondrial domain of the protein that enables the clustering on the cell surface. And this is that 271 position from before that, when cut, separates signaling from localization. And then this 93 position is the one I'm talking about before. So this variant here early on we ignored because the changes in 1,000 is registered in 1,000 genomes, and it was a common polymorphic variant in the human population. The glutamate allele, the E allele, which was the one that we were working with, was rarer overall and rather rare of those in African descent, for example. But it was just as prevalent as glutamine in those of East Asian descent. And so we thought that this was just a natural variant. Yet the glutamine is the sequence that's widely conserved in mammals, and its presence creates a second potential cleavage site for 3-seed proteinase. Again, you're getting one of these requisite glutamines in the consensus sequence. That would also separate assembly from localization, just as that prior 271 site would. And so just to finish up here, we wanted to try to test this using the exact same things that I talked about before. We make the mutant, stick it into the cells. Compared to the wild type, we show increased cleavage at the position when we change to the glutamine. And then the next step was impact its overall effect on infectivity. But before showing the result, I want to explain what we thought that the result might be. We put in a glutamine substitution. Now we have 2 cleavage sites on MAVS. We thought that by being twice as likely to be cleaved, the glutamine allele will be less potent as an antiviral signaling molecule compared to the original MAVS, because it'd be twice as cuttable. Wrong, and not only wrong, interestingly wrong, because this glutamine allele was even more potent than the MAVS that we started with. And even more confusingly, it also reduced cleavage on the other site, the one that we were reading out is this 35 kilodalton product. And so here we needed to pivot, kind of like in over our heads, needed a different abstraction that would focus specifically on the behavior of MAVS. And unlike this complete kinetic model that I described before, this model was focused only on connections and feedbacks within MAVS regulation, and less concerned with the specific parameter values that I dwelled upon with that complete kinetic model. So we built this state model in which MAVS is synthesized or activated, it doesn't matter in this state model, and assumed to self-assemble rapidly on the mitochondrial membrane. The filament size here, based on the literature, is large. They form these 600 to 800 MERS. And that assembly was assumed to be instantaneous very fast because it's on a membrane surface. And it's those MAVS filaments, those aggregates, that they're degraded by an autophagic route, but they also lead to signaling through the production of ISGs that themselves have a degradation rate in this state model. And so when there's no cleavage in this simple simulation, MAVS and the polymerized MAVS reach steady state values you can solve just like you would in differential equations class. However, when we incorporate the steady state production of a 3C proteinase, like during an active infection, and one cleavage event corresponding to that original MAVS that I started off with, there's an additional route to MAVS elimination from the system, and then the production of interferon-stimulated genes is not sustained. We include negative feedbacks here for completeness, but it turned out in the model they were not critical for the properties I'm describing here. Interestingly, when there were two separate routes to MAVS degradation, as with that polymorphism that I was describing before, we see different behavior with now sustained interferon-stimulated genes, as shown in the brown here. And that flat brown polymavs trace here on the right isn't a steady state, but it's a slowly decaying exponential when the amount of monomeric MAVS here gets small relative to filamentous MAVS. So when that starts to get small and decay away, you just get a first order decay rate with this time constant. And as I said, N is 600 to 800, which really drops down the rate of decay. And that polymorphism reaches this decay regime 4 times as fast because of those two routes to degradation. So this alpha parameter here that dictates how many times you can get cleaved, going 1 to 2 hits the peak here of MAVS polymerization twice as fast and then troughs twice as fast, meaning overall 4 times faster to reach to the state and then that longer decay. So what it converged toward here is that the current understanding of MAVS in this very simplified model is sufficient to explain the counterintuitive behavior of its variants. And so just to finish up, I want to touch on one implication for this for antiviral disease, that human variation in MAVS may have important for infectious disease, because recall that that single cleavage variant has a prevalence of about 50% in the East Asian population. What that implies is that 25% of East Asians are homozygous for the more virally susceptible single cleavage variant. And East Asia also struggles with endemic enteroviral disease, which outbreaks every 3 years or so. And that peak incident occurs among children 2 or 3 of age. And so it's tempting to speculate that these cycles might relate to building up enough virus-naive homozygous individuals in the population to foster community spread. And so I'll close by acknowledging the students who assisted with the work that I showed. You see it was a multi-generational effort. And then I'll point you to the bioRxiv preprint of this work. And then if you're interested to learn more about my group or my department, I leave these websites up for you. Thanks, and I'd be happy to take any questions.
That was beautiful work. I think we better move on to our second talk, and then if there's time, have questions for both talks at the end.
Sure.
Because I apologize, but we have to accommodate 2 speakers.
Understood.
And I want to make sure that everybody has time. I'm sure there will be questions, and there'll be plenty of opportunity to answer questions offline as well. So I'd like to move on to our second speaker, Rahman Sherriff, on reproducibility and systems biology modeling. Are you queued up and ready to go?
Yes, thanks.
Okay, I will turn it over to you.
All right. Okay. Can you see my screen? Perfect, thank you. So thank you for giving me a slot to speak today. So the title is slightly different. As you can see, it's the reproducibility in systems biology modeling and biomodels role. So in this talk, I'm going to talk first about biomodels, what it is about, and then about the study that's done in biomodels. Right. So what is biomodels? For those who don't know, biomodels is a database of mathematical models of biological processes. It was established about 15 years ago as a platform for modelers to exchange models with each other. Right. If you're a modeler, you can go to Biomodels. You can submit your model, which can be in any modeling formalism. It can be ODE, logic, Petrinet, any model, constraint-based model, and they can all be submitted in to Biomodels. And these models can be in any format. They can be a MATLAB script or a C or Python script or standard such as SBML, SLML, and so on. And these models, upon this submission to Biomodels, are searchable and can be found by other modelers. So one advantage of submitting a model to Biomodels is the curation. So models submitted to Biomodels are carefully checked by the curators in my team. So by the way, I lead Biomodels team, and they compare the equations in the publication of that manuscript, the manuscript describing the model, and they encode the equations in computer readable standard formats such as SBML. They run simulations using open source tools and try to reproduce the simulation results. If a model is reproducible, if it does what it's supposed to do, and if the simulation matches what is described in the publication, the simulation figure in the publication, then we stamp this model as curated and reproducible. This is very good because then the users who come to buy models can look for models which are verified. And it's also good for the modeler who submitted the model because there is some external verification or validation of this model. And we have seen that users tend to download these verified or curated models twice more than the non-curated models. Okay, so it's not just the models from the curators, but we also do targeted curation. We try to find certain area of research and then try to curate more and more models and add it to the Biomodels database so that modelers can benefit from that. Currently, we are focusing on curation of COVID-19 models. We have good collection of COVID-19 models, and if you can go to the link shown below in this page, you will have a good collection. All these models are open source and it's free to use, so anyone can download them and reuse them. Okay, and we have many more collections. So if you go to Biomodels, you can search your keyword of interest, immune response or COVID-19. You'll find a list of models, and then you can filter them based on the facets on the left-hand side and narrow down your search and find the model of your interest. Once you go to the model, you can see a title of the model and then the manuscript. You can go to you can click the link and go to the publication, or you can also find some quick overview of this model. What is it about? Which organism it is. Like on the right-hand side, you see some metadata saying that it's for humans, Homo sapiens, and it's about T-cell regulation. So we add extra ontologies and control vocabularies to make these models more semantically enriched, and so they are findable by search. And also gives meaningful information for the modelers who try to reuse or repurpose these models. Okay, moving on, Biomodels is widely used. In a survey of scientists, many of them said that they go to Biomodels because it's one of the important data source for them. And also many modelers prefer to submit their model to Biomodels after supplementary material, GitHub, and personal website. As I told you, this is again because of the curation services probably, and also the models are more findable when it's in such a repository. Okay, so you can read more about our work here from these publications. I will move on to the next slide quickly. Okay, now coming to the main topic of the discussion. Reproducibility in systems biology modeling. So before we go there, we need to understand the terminologies so that we are on the same page. So replicability or repeatability is the ability to use the same code provided with the manuscript using the same software to reproduce the simulation results. Yeah, but reproducibility is the ability to recreate a model without original code. So replicability or repeatability is using the same code, using the same software, reproducing the simulation. But reproducibility is the ability to recreate the code without recreate the model without the original code. So in our study, we focused on the latter, that is the reproducibility of the model. So we assessed reproducibility of 455 models from published peer-reviewed articles and tried to find how well they are reproducible. So this is one of the largest study ever done to assess the reproducibility in this field. Right. So what did we do? We encode the models in SBML, and we get the parameters values from paper, supplementary material, put them all or from the authors and put it together. So why do we use SBML? SBML is one of the standard community standards used because it's highly interoperable, if you have a model in SBML format, it can be imported into several softwares, over about 300 softwares. Tools support SBML. So if you can import it into MATLAB or Python or R or Java. So you have huge flexibility. So when you share your model in this sort of community standards, you can import them in several formats and reuse them. Okay, so we encode the model in SBML. We run the simulation using open source tools such as Copasi, and then we try to see if the figures are reproducible. So we consider a model as reproducible if at least one simulation figure from the publication could be reproduced. Then we are happy. We say this is reproducible. So we still we are not very strict. We accept minor deviation if it doesn't affect the scientific conclusion of that study. If a model is not reproducible, we try to fix it. Let's say there could be some error in the equation or parameter. We can try to play around and tweak the model and empirically try to fix the model. If we still can't fix it, then we try to contact the authors and get the information from the authors and try to fix the model. So now the results. Before that, give an example. Yeah. Sorry. So let's say you have publication with certain simulation figures for the model described in that manuscript. We reproduce it, and this is part of the curation which you do in biomodels, and this figures are reproduced and added to the curation tab of that model in the biomodels. So you can cross-verify that these models are indeed verified by someone and reproduced. Okay, so in this study, we have curated about 455 kinetic models, that is ordinary differential equation models of biological processes. These are from peer-reviewed research articles from 152 different journals published from 1980 to 2020. So this activity was done as a part of normal biomodels curation, and hence the models are not randomly sampled, but they are selected based on the priorities of curation in biomodels. So this 455 models are a significant proportion of models available in biomodels. Okay, now the results. Sorry, now here is the main results. So we would expect that these mathematical equations are reproducible, but unfortunately, only half of the models are directly reproducible. Half of the models could not reproduce based on the information provided in the manuscript. Of course, this was of course surprising and also not, because we have heard from the community always that there are models which do not reproduce, but no one knew how much prevalent is this. And from our study, we see that at least half of the models are not reproducible directly based on the information provided in the manuscript. Okay, so those models which are not reproducible, we went and did put some additional effort. As I mentioned, we tried to do empirical correction. We tried to tweak the model, look for typos, any mistakes in the equations, or any typos in the parameters. It could be 0.1 instead of 0.001. We try to play around a bit and see if we can fix the model. Yeah, and by doing that, we almost fixed 9% of the models in total. And then we try to contact the authors, and from the authors' support, we kind of reproduce 3% of the model out of 455 models. And finally, 37% of the model could never be reproduced. Yeah, we could not reproduce based on information provided or from even additional effort. Right. So when we tried to contact the authors, it was not an easy task. Of course, there were publications which were very old. We could not even reach authors, but then we still managed to contact about 90 authors, and majority of them, 70% of them, do not respond. Yeah, even though it's good for them to have the model somewhere in a public domain, open for openly visible, they did not respond, and only 30% respond. Yeah, even in those 30% who responded, we could only reproduce half of the models. So half we could not reproduce. Sometimes they respond and they say, oh, right, my PhD student is not here anymore. I cannot find the code and so on. So we simply can't reproduce even after we contact them. Even sometimes they provide some information and that's not good enough for us to reproduce the model. Okay, moving on. So if you look at the age of the model, there's no indifference. This is the similar trend. If you can see the proportion of the model that are reproducible, not reproducible, more or the same across all the years that we have analyzed. And so you can see that only we could contact the authors in the last 10 years, not before. Okay. When we also looked at the journals and see if there's any clear pattern, if some journal is better, some journal is bad, we can't really conclude anything because few journals had lots of publication with mathematical models, and of course, a few journals had less models. So there's no clear pattern in terms of journals on the reproducibility of the pattern. We did compare the size of the model, the total number of species in the model, and what we found is that smaller models, of course, more reproducible, and bigger models are more, difficult to reproduce. But still, a significant proportion of the small models are not reproducible as well. Okay. So the reasons why these models are not reproducible, we try to look into the reason. Of course, as we know about a lot of models were not reproducible due to missing parameters. They did not report a parameter value in the manuscript, and sometimes did not report initial condition. Sometimes we try to guess from the simulation figure. Of course, it's not possible to guess all the missing values for initial condition, and sometimes there is some error or in the equation of the model and so on. Okay, and for still a huge majority of the model, like 70 models, we cannot find any reason. There's no reason that's obvious to us, at least. So we try to explore, but there's no results. So that could be something wrong. It could be a wrong parameter, wrong initial condition, wrong equation. No one knows. Okay, so where are these models available? So all these models are now in biomodels. So those which are reproduced or under the manually curated section of the biomodels, and those models which are not curated, not reproducible, are under the non-curated sections of the biomodels. So in the non-curated section, you cannot consider all models are bad, because this has models which are which fail to reproduce, and also those models we didn't have time to look at, and also those models or with the modeling formalism we are not yet curating. But we are under discussion to tag these models which failed as as failed models, or some sort of labeling, and we discussed with the community, and most of them gave us feedback that there should be some sort of label to indicate that those models which are failed should be distinctly labeled, and the reason for failure should be labeled. So we are in process of doing that. So hopefully we'll do that soon. Okay, so what is the solution? So we had proposed like an 8-point reproducibility scorecard, which can be used by authors or editors and reviewers to assess the reproducibility of the models. So these are the 8 questions we have here. So are the mathematical expression described in the manuscript, or are the parameters and initial conditions given in the manuscript? Are the changes in the simulation conditions are described in the manuscript, the normalization described in the manuscript? Are the model codes shared publicly? Are the codes available in a community standard format, or the codes in relevant database? Are there annotations or semantic enrichment in the model? The numerical results shared publicly in these models? So we came up with this 8-point scorecard to assess the reproducibility. We say that this can be used by authors, reviewers, editors to assess how reproducible a model is going to be. So is this a good indicator? So yes, we try to do some extra work. We picked up over 110 models and tried to calculate the reproducibility score for each and every model and ask if these are reproducible. So we set a cutoff. We propose a cutoff of 4 or more. Of course, not all models have all the tick all the criteria. So we found that if a model has got a score of 4 or more, it's highly likely to be reproducible. Yeah, you can see that our odds ratio is 4.62, and from these 110 models which we analyze. So this is the solution which we think is proposed, and this is what we recommend modelers to use to assess reproducibility of the model. Okay, last slide now. So this work is now, it's been there out for a long time. It's in bioRxiv and is now accepted in molecular systems biology. Hopefully it will be out there soon as well. It's quite tweeted and discussed in the community as well. And hopefully there will be some positive impact in the community, and there will be more and more reproducible models in the field. All right, with this I would like to end the discussion and acknowledge all the team members who to help to curate the models and the funding agency, which is IMI, and then BBSRC and the EMBL. And thank you for your attention. Of course, in the context of a subgroup, I'm co-leading the reproducibility group with Jacob. And if you're interested in reproducibility, I welcome you to join the subgroup. And I'm happy to take any questions.
Okay, thank you. So we have time. We have a strict cutoff at 4, so we don't have too much time. Reinhard, was there any business that we needed to urgently go through, or shall we have some time for some questions?
No, I think we have time for some questions. I think if there's time left, probably you should discuss your ideas about competitions.
Okay, so thank you to both of our speakers for very interesting talks. And I'd like to open the floor for 1 or 2 questions to our speakers.
So I have a question. Go ahead, Fred.
I was going to try to combine my 2 questions. I'd asked Kevin, thanks for that great talk, by the way, about stochasticity. And for the second talk, how do you deal with stochastic models and reproducibility?
Who's going first?
Kevin, I think you should go first.
Okay, I can go. I can say a little bit like the way we have some element of stochasticity rudimentally encoded in the very beginning is in the Poisson, like how many virions actually land on a cell. But beyond that, one of the points that we struggle with, which I glossed over, is if you have one virion that successfully docks and delivers into the cell, where does this continuum approximation hold water anymore to go on? And so I get in the inner workings, the way that we do this is we kind of run, we don't formally simulate the stochastics, but we don't read into any of those early simulations up until you get this critical threshold where what happens is that these RNA viruses steal membrane. And we basically right now arbitrarily set like 25 copies of this critical molecule that steals membrane. And then the architecture flips around and now the replication starts happening on a surface instead of in solution. And then that's where we say that there's an appropriate continuum regime approximation, and then we pay attention to those. But actually that switch is fascinating because we can only encode it a handful of ways to get all the nice calibration. There's other architectures that you think would work and it doesn't, it's much noisier. There's not anywhere near the reproducibility. And I actually think that might be saying something about what the membrane stealing might have an evolutionary advantage for this category of viruses. So I'll stop on that.
Okay, coming on to the next question, how are we dealing with stochastic models? So in this study, we just focused on OD models for the sake of simplicity. And as I mentioned, so we do not want entire figure to exactly match, but as long as it reproduces simulation that doesn't change the meaning of the study, we accepted that as reproducible. So probably we apply the similar criteria there. So we don't expect one-to-one precise match with certain tau or precision here. Probably something similar will be applicable here.
So I have a question. First of all, there's great work for both of you. Thank you very much for both talks. Roman, so the results of your study are really depressing, right? So I'm wondering if so you developed these 8 rules that you postulate as a guide to reproducibility. Would you recommend that any journal that takes papers like this should enforce these rules?
Exactly, yeah, exactly. That's what we would recommend. So we kept it simple. There were other solutions. There were, some sort of additional curator-based evaluation of the models and so on. So, but we want to keep things very, very simple. So these points are very easy to evaluate. You can just have a checkbox and check them. And then, as I showed in the stats, then if the model has score of 3 or 4 or more, it's highly likely to be reproducible. So we will slowly start communicating with the journals and recommend that they use this this criteria to assess the models whenever there's a publication with model. Yeah, definitely, we will do that sooner.
Great, thanks. And just to follow up on Fred's question, you mentioned in the beginning that you also take things like agent-based models. What do you do with them?
Oh, so Biomodels as a database for sharing models, we consider all modeling formalism. So you can share and exchange models, but we haven't yet touched on agent-based models because we don't have expertise on that field. But then hopefully we will gain soon. So initially, we have been only looking at OD models. Now we have expanded into constraint-based models. I'm in discussion with this working group, which is trying to develop standards to assess the reproducibility of the constraint-based models. And there's also this discussion of the logic-based models. So we are expanding slowly at the moment. So biomodels has been initially known as Kinetic model repository, but now we will have soon curation done for these two. And hopefully in future, we will also do assessment of agent-based models.
Great, thank you.
So Rahim and John Rice, my concern is dissemination of useful knowledge coming from computational models to, let's say, a clinician. How do what is it I need to explain to clinicians about reproducibility as a characteristic of a model that they should be aware of and that would help them have some better trust or confidence that maybe a computational model just might be useful? How do we communicate that to people that we want to have using the output of models so that it becomes part of their knowledge of modeling and why they might be willing to accept models as useful?
I think, John, you asked a very good question. It's a very important question. I think there is no clear answer for that. But I think as in any experimental finding, reproducibility is very important, and it's important for us to convey. Instead of, for the clinicians, maybe first thing we need to convey is that we need to emphasize how important is or how useful are these models for them in their work or in their research and so on. So that could be the first step. And then the next step is, of course, making sure that they choose the models that are reproducible. So similar to the fact that they will prefer a study which is more reproducible for implementation or for any kind of clinical activity around that. So similarly, they should also pick up a reproducible model.
Yeah, but I think that at some point it's going to become very useful that people with your expertise and experience and like the examples that you were able to show could write the short kind of article paper that is written as though you were going to go in and teach a physician.
Yes.
That this information we're trying to give you came from a reproducible model. What does that mean to you? How do you explain that? I would love to see just a rough draft of a paper like that. And then we can figure out how we get it to people or make it better or whatever. Thank you. That's it. Yep.
Yeah, sure. Definitely. Thanks for the comment. I think I'll be interested. Yeah, definitely. This will be very useful.
I apologize. I don't want us to time out. Is there any more questions for Kevin quickly? We have time for one minute question. Jacob?
I have one.
1 minute.
You use differential equations, and Fred was asking you about stochastic simulation. Did you try agent-based modeling, microsimulation, any one of those techniques? Are they useful, and where do they fail if not? And I'll be quiet now.
Yeah, so shorter answer is we didn't, because it took us 5 years to get the continuum model to match to our experiments, which weren't single molecule. But now I think we can start looking into, I think what's clearly tricky is at the end, you're dealing with millions and millions of individual molecules. And so that transition from a discrete stochastic to continuum regime is a tricky one to sort of navigate. But we have a reference point now to feed that in.
Talk to me offline.
Oh, terrific. Excellent. Thank you.
So Kevin, John Rice again, I'm gonna press my luck and give you the same question. It's like, I love what I think I understood you showed. What would that look like if it had no formulas in it? And you were setting out to go out and sell a physician on what you just show as a factor in how they treat patients.
Mm-hmm.
Could anybody write that lecture?
Yeah, well, and so I think that I was actually gonna weigh in on your prior question. Step one for one of the things that I think is a key aspect is user friendliness. I mean, the fear that most clinicians or basic scientists have in the command line is just, that's a non-starter for people. And so for us, like this model is out on GitHub and things, and we basically just say, type this in and it will run.
Yeah.
README. And that's even not enough for virologists. And so it's next phase for us is I have a proper virology postdoc in my lab. We're going to do a graphical user interface where it's clicks and slider bars and just the graphs come up and go, because all of that makes it really reduces the activation energy barrier to adoption and experimentation and playing around with it to get some intuition about what it does and how to interpret it.
Now, as this big working group forms up and these weekly meetings are phenomenal, that from the perspective of a dissemination supporting group within it, I would love to talk to you and a couple people offline on a Zoom about for demonstration purposes, what could we put together kind of quickly that we could actually take to a few MDs and sit down and say, try this. And because eventually,
Done.
Yep.
We're out of time.
Sorry. Okay.
But Kevin, if you want to put it up on a server to do exactly that, I can do it in an afternoon with you.
Fantastic.
It's literally a 2-hour process.
Great. Yeah. Like I said, I have a postdoc who's enthusiastic about these sort of things. And so absolutely. I mean, he knows how to do the model and everything.
Thank you, everyone. I know these talks excite enormous amounts of interest and the format doesn't let us go through them. Please connect with each other offline. And I hope that these two very stimulating talks give rise to lots more. Again, our speakers today are welcome to come back and give follow-up talks. Look forward to seeing everybody again next week. We have a steering group tomorrow. So those of you who are on the steering committee know who you are. Please join us. And some of the material, the business material that we didn't get to talk to today, we'll talk to tomorrow. Thank you, everybody, for joining us. Thank you very much for our speakers and your patience in trying to squeeze things into a short amount of time. I apologize for not having more time for conversation and questions. Thanks, everybody.
Thanks very much. It was a lot of fun. Take care.
Bye.